A genome browser is an online graphical interface used to display genomic data. The information needs to be stored in a database so that it can be shared with lots of people around the world. Whereas the sequencing the whole genome allows research to study not only genes which code important proteins, but also regions of our DNA that have other important roles. Hello everyone, this is Rishabh Sopar, the student of BTech Bioinformatics, and I'm going to represent Genome Browser NCBI. Before we get started, let's talk about some highlighted main components, which is methods, search function, sequence viewer, exon navigator, subjects, and genotype table. But introduction first. In bioinformatics, a genome browser is a graphical interface for display of information of a biological database for genomic data. Many genome browsers are available, many of them free and with database accessible online. Among the best known are the UCSC Genome Browser, ANSYS Genome Browser, and NCBI's Genome Data Viewer. Here is a method. Method to open the Genome Browser. Open the Chrome web browser or whatever you want to use to open Genome Browser. You can directly search of this given link for open direct link of the Genome Browser. This is the main interface of Genome Browser as you can see on your screen. There, there is lots of components and functions out there which we use to study genomes easily and compare also. Search function. First, here is a search example. Under the search box, we see the search examples. It shows gene symbol, phenotype, variant IDs from dbSnip or dbVar, and varieties of formats for sequence coordinates. Example, we are going to use RS1008-407 dbSnip ID, then hit enter. Now let's talk about sequence viewer. The sequence viewer is just a screen or display portion in a genome browser where the exon sequences and all the other results are shown. The SNP is located into gene which is OCA2. The sequence viewer is centered on a SNP displayed as dark green. Also changes the tracks menu. We can also record the tracks, zoom and paint the tracks and CBI resources by clicking upon the Exon bar. Now let's talk about the sequence viewer in a detailed way. The SNP is located into OCA2 gene. It turns one allele of gene into transparent blue. The sequence viewer is centered on SNP displayed the dark green region as an exon of OCA2 gene, as we can also change the display tracks by tracks option menu and also record the tracks, zoom in and zoom out tracks, and pane to open some menu with additional information from the another NCBI resources by clicking upon the Exon bar. By hovering the cursor over the marked SNP brings the useful menus like marker details. It carries the position of markers as genomic, transcript, protein, and coordinates with notations HGVS. Here as you can see the sequence viewer look like. Now let's talk about the exon navigator. Exons represented by the outlined blue colored circle bullets. It displayed the coordinates. We can adjust the displayed sequence. The genotype table updates the variation displayed into the sequence browser. How to align the sequence in sequence viewer. Just expand the population row, select genotypes for individual samples. Check the box of sampled row of Exon. It shows the alignment on header. Matches show gray, whereas the different will be red in color. Now let's talk about Exon Navigator in a detailed way. The current exon is the outlined blue-colored circle. By clicking on it, the boundaries of exon in Screen Viewer and display the coordinates of exon under the circles. To zoom out the boundaries of certain gene, use region menu and select an option for switching between genes displayed region using gene menu. For switching transcript by transcript menu, we can adjust the display sequence. The genotype table updates here. Each table represent coordinates. White columns correspond the variant's current display into the screen viewer. Blue column are selected coordinates and snip in the case. First header shown tooltips with various display from 1000 genome project. Second rows shows the global allele frequency. Other country name rows show a specific population allele frequency. Here show how to show the alignments in Screen Viewer. Expand the population row here genotypes for individual sample. In population, click and check the box of sample row. That's exon. It will show the alignment on header. Now let's see about the aligning the sequences. This is aligning of sequence how look like. As we know from previously, there is the match show gray, whereas the difference will be red in color, as you can see on your screen. Now let's switch to the genotype table. This window is known as the genotype table, as you can see on your screen. The genotype table carries the thousands of different alleles from the different regions. From here, we can easily select the sequence for comparing with other sequences and alleles. Now let's switch to the subjects. Choose alignment tracks from the subject, check the box, and update to add into the display. We can also choose the alignment tracks from the subject section. The available alignment and filter tracks are in the section of window. Here we filter or search the specific tracks, then click on the checkbox and update to add into the display. Genotype. And last into the download section provides an option to download the SRA reads or the displayed region. Thank you and thanks for watching.